Hello, everyone. Uh, can someone assure me that I'm audible? If I'm audible, you can either unmute yourself to confirm this or just give a thumbs up. Okay. Thank you, Rakshana. Thank you, Narendra. And it's clear also, right? Because I'm not using any headphones or something. So I hope my voice is clear to you. So we'll start our session within like 3-4 minutes. I'm expecting more participants to join as this is our first live session for animal physiology course. In the meanwhile, when we are waiting for other participants, if you all are comfortable, you can unmute yourself one by one and like put your videos on and have a little introduction, right? So that we know to whom I am interacting with and who you guys are, correct? So if you guys are comfortable, we can go ahead with this thing. Otherwise, every one of you can put your basic introduction, like your name, your course, which you are currently pursuing, from where you are pursuing, and from where you are, guys, right? Those basic information you can send in the chat box if you're not comfortable unmuting and turning your video. Also, I will apologize in advance. My throat is not well, so I might cough in between, and I'm sorry for that. So, yeah, if you guys are comfortable in turning your videos on, just turn your video on, and I'll call one by one to give your basic introduction, and then I'll tell you about myself. So let's make this session interactive, because all the sessions which I'm going to conduct will be in interactive mode only. I'm not going to teach you guys anything because that is the work of our NPTEL videos, lectures by Professor Manik Das. I'm here to clear your doubts, certain doubts, and help you by solving some previous year assignment questions to make sure that your current assignment, it might help you in solving those, right? So, oh, no one is comfortable in uh, turning their video on. Okay, it's all right. So you guys can drop your basic introduction in the chat box, your name, your course which you are currently pursuing, and year also, from where you guys are pursuing that, and last, from where you guys are. And if any fun fact you want to add in it, feel free to add in it.
Good evening, madam.
Hi, Narendra. Yes.
Yeah. Am I audible?
Yes, very loud and clear.
Yeah. Thank you.
Hmm.
Yeah. Actually, I have recently joined this course, Animal Physiology.
Okay.
Like I got two mails saying that session 1 and session 2 at 5 to 7 and 6
Six.
to 8.
Yeah.
So I'm just confused which one to attend.
You see, there are basically 3 TAs. I am a TA for this course. TA means teaching assistant, right?
Okay.
So for
Okay.
this particular course, we have, I guess, 3 TAs, which means
Okay.
in a week, you will have 3 live sessions. All
Okay.
these live sessions will be similar in nature.
Okay.
The
Okay.
way of solving these questions might change, but the
Okay.
topics which will be covered, it will be same. So you're
Okay.
not leaving anything behind. It is
Okay.
just a convenience of time.
Okay.
Like whichever session you are comfortable with, you can join.
Okay, any one session we can join.
Yeah.
Yeah,
Because we're gonna discuss similar questions. It's
right,
just way, because there will be a particular way I am explaining things, right?
right.
Some
Right.
other person will explain in different way. So
Yeah.
the things will be same. It's
Mhm.
just the timings are different and the TA will be different.
Okay,
So you can attend any session according to your convenience.
right.
Yeah.
And one more doubt, madam.
Yeah.
Actually, I'm also registered up for another course.
Yeah.
So even that course also scheduled in this same time.
So there might be at least 2 sessions for it in a week, maybe.
Yeah,
Yeah,
correct.
so then you need to work around.
Oh, that
Because
is, yeah.
see, if you're for example, if you're comfortable in attending 6 to 8, right, so that you
Hmm.
can attend other courses live session also, you can do that. And I'm not aware about the third session for this course, like when it is scheduled. I'm not aware of that. I'm
Okay,
aware
okay,
of Saturday course. Mine is from 5 to 7 and other is from 6 to 8.
okay, okay, okay.
You have plenty of options,
Hmm.
at least, like for example 3 TAs for this course, and for the other course also you might be having at least 2 TAs which will be taking session. So two
Okay,
TAs means in a week you will be having 2 live sessions.
okay,
Three
okay,
TAs means in a week you will be having 3 live sessions.
okay,
Yeah, so you can select anyone according to your convenience.
okay.
So
Right.
now you need to introduce yourself because you unmuted. Yeah.
Okay. Yeah, thank you.
Ah,
Myself, Narendra Reddy. I'm a bachelor. I did my master's in biotechnology from
cool.
Usmania University. Uh, right now, I am working as a senior scientist too in Vimta Labs Limited as a toxicologist. I'm
Oh,
having 13 years of experience in the field of toxicology.
I should call you sir then.
No problem.
So, like, what motivated you to attend this course?
Actually, I'm taking a part-time PhD program. So almost 8 months, 1 year back, I have joined it.
From?
So from Vignan University,
Okay. Which department and
biotech,
specialize? But
biotechnology. Yeah.
okay. So your lab currently works in what section?
It is not the lab working is a different field, but I will do my PhD work in same field where I am working, toxicology
Oh,
stream only.
okay, okay, okay.
Yeah.
Great. So, do we have any other active participants who would like to introduce themselves? Guys, why are you doing this? It's a like, and it's completely not official, right? It's completely fine. You guys can be very frank with me.
Correct. Yeah.
You are also learning with me. I'm also learning with you guys. So uh be open about discussions. And because I'm not going to teach you anything new here. The only thing is I might help you in understanding how to solve assignments or how to look for problems when they are there, and you might not know what is the correct answer, but there are certain ways through which you can get to the right answer, correct? So those things and new concepts, I might not be introducing new concepts, I might be clearing the concepts which have already been taught in the like lecture videos. I hope all of you have covered the first week lectures, right? Just give a thumbs up if all of you have covered first week all the lectures.
And one more doubt, madam. I think this is the first week.
Basic information you can send in the chat box if you're not comfortable unmuting and turning your video. Also, I will apologize in advance. My throat is not well, so I might cough in between, and I'm sorry for that. So, yeah, if you guys are comfortable in turning your videos on, just turn your video on, and I'll call one by one to give your basic introduction, and then I'll tell you about myself. So let's make this session interactive, because all the sessions which I'm going to conduct will be in interactive mode only. I'm not going to teach you guys anything because that is the work of our NPTEL videos, lectures by Professor Manik Das. I'm here to clear your doubts, certain doubts, and help you by solving some previous year assignment questions to make sure that your current assignment, it might help you in solving those, right? So, oh, no one is comfortable in uh, turning their video on. Okay, it's all right. So you guys can drop your basic introduction in the chat box, your name, your course which you are currently pursuing, and year also, from where you guys are pursuing that. And last, from where you guys are. And if any fun fact you want to add in it, feel free to add in it.
Good evening, madam.
Hi, Narendra. Yes.
Yeah. Am I audible?
Yes, yes. Very loud and clear.
Yeah. Thank you.
Mm.
Yeah. Actually, I have recently joined this course, Animal Physiology.
Okay.
Like I got two mails saying that session 1 and session 2 at 5 to 7 and 6
Six.
to 8. So
Yeah.
I'm just confused which one to attend.
You see, there are basically three TAs. I am a TA for this course. TA means teaching assistant, right?
Okay.
So for
Okay.
this particular course, we have, I guess, three TAs, which means
Okay.
in a week, you will have three live sessions. All
Okay.
these live sessions will be similar in nature.
Okay.
The
Okay.
way of solving these questions might change, but the
Okay.
topics which will be covered, it will be same. So you're not leaving anything behind. It is
Okay.
just a convenience of time.
Okay.
Like whichever session you are comfortable with, you can join.
Okay. Any one session we can join.
Yeah.
Yeah.
Because we're gonna discuss similar questions. It's
Right.
just way, because there will be a particular way I am explaining things, right?
Right.
Some
Right.
other person will explain in different way. So
Yeah.
the things will be same. It's
Mhm.
just the timings are different and the TA will be different.
Okay.
You can attend any session according to your convenience.
Right.
Yeah.
And one more doubt, madam.
Yeah.
Actually, I'm also registered for another course.
Yeah.
So even that course also scheduled in this same time.
So there might be at least two sessions for it in a week, maybe.
Yeah,
Yeah.
correct.
So then you need to work around.
Oh, that
Because
is, yeah.
see, if you're for example, if you're comfortable in attending 6 to 8, right, so that you
Hmm.
can attend other courses live session also, you can do that. And I'm not aware about the third session for this course, like when it is scheduled. I'm not aware of that. I'm
Okay,
aware
okay,
of Saturday course. Mine is from 5 to 7 and other is from 6 to 8.
okay, okay, okay.
You have plenty of options,
Hmm.
at least, like for example, 3 TAs for this course, and for the other course also you might be having at least 2 TAs which will be taking session. So two
Okay,
TAs means in a week you will be having two live sessions.
okay,
Three
okay,
TAs means in a week you will be having three live sessions.
okay,
Yeah, so you can select anyone according to your convenience.
okay.
So
Right.
now you need to introduce yourself because you unmuted. Yeah.
Okay. Yeah, thank you.
Uh,
Myself, Narendra Reddy. I'm a bachelor. I did my master's in biotechnology from
cool.
Usmania University. Uh, right now, I am working as a senior scientist too in Vimta Labs Limited as a toxicologist. I'm
Cool.
having 13 years of experience in the field of toxicology.
I should call you sir then.
No problem.
So, like, what motivated you to attend this course?
Actually, I'm taking a part-time PhD program. So almost 8 months, 1 year back, I have joined it.
From?
So from Vignan University,
Okay. Which department and
biotech,
specialize? But
biotechnology. Yeah.
okay. So your lab currently works in what section?
It is not the lab working is a different field, but I will do my PhD work in same field where I'm working, toxicology
Oh,
stream only.
okay. Great. So, do we have any other active participants who would like to introduce themselves? Guys, why are you doing this? It's a like, and it's completely not official, right? It's completely fine. You guys can be very frank with me.
Correct. Yeah.
You are also learning with me. I'm also learning with you guys. So uh be open about discussions. And because I'm not going to teach you anything new here. The only thing is I might help you in understanding how to solve assignments or how to look for problems when they are there, and you might not know what is the correct answer, but there are certain ways through which you can get to the right answer, correct? So those things and new concepts I might not be introducing new concepts. I might be clearing the concepts which have already been taught in the like lecture videos. I hope all of you have covered the first week lectures, right? Just give a thumbs up if all of you have covered first week, all the lectures.
And one more doubt, madam. I think this is the first week's lecture, no?
Yeah.
Then I did not attend any other session prior to this. That's why.
No, this is live session. This is for
Okay.
problem solving. But
Okay.
videos were already live in the NPTEL portal, right? Where you have registered for the course. The first week lecture videos were already present, like 5 lecture videos,
Yeah. Okay.
right?
I
So I'm
did
talking
not
about those.
actually. I did not access for the means. I did not see those videos till now.
Oh,
So
so this is just a problem solving session. I'm not going to teach anything. So you
okay.
go back to your portal where you have registered for this course,
Mm-hmm.
right? You will have and I guess now week second lectures are also online. So you will be having week 1 lectures, which will be having, I guess, 5 videos in it and an assignment associated with first week. Then
Okay.
you will have second week lecture videos, which are again, I guess, 5 or 6 in number and an assignment associated with it.
Okay.
So these sessions are for helping you in solving those assignments, which are weekly assignment already
Okay.
posted in the NPTEL portal.
Right. Okay. Now I got.
Yeah, it's okay, no problem.
Yeah.
And kindly unmute, and if you want to ask question, just raise a hand. Yeah. Narendra, can you mute yourself if possible? Yeah. Thank you. Thank you. So, okay, how? Hi, Priyanka. Good evening. Uh, I think so that I might butcher your name, but is it Samadrito Majumdar? Okay, so you are Presidency University graduated. Great. But others also, like I can see there are plenty of people, right? At least drop your name and the course which you are currently involved in. Then only I will know na, how to teach you guys, right? Otherwise, how will I be able to teach you? Yeah. So, hi, Shweta. Hi, ma'am. I'm Shweta from Tamil Nadu. Okay. I'm second year student of biomedical engineering. Great. Okay. Hi, Zafira. Human physiology, and I'm doing B bio. Great. From where you guys are doing your courses? Swetha, from where are you pursuing your course? And Zafira, also from where are you attending your BE in biomedical engineering? Hi, Mansi. From where you are attending BTech Biotech, right? It's a very basic introduction. Don't be scared. I'm not your professor. Trust me, I'm not. Right? Hi, Gauri. And I'm currently pursuing BSc Microbiology. Oh, great. We have diverse students. I like this. Hi, Aditya. BSc Biotechnology from Amity University, Ranchi. I'm also from Amity University, but from Jaipur. I did my BTech from there. Hi, Amali. Okay. Yeah, from where are you? What course you are enrolled in? Not this animal phys. I know that you guys are attending animal physiology. That is why you are attending this live session. I mean, what degree you guys are pursuing and from where you guys are pursuing. Hi, Xavier. I think I will butcher for your first name, so I am not gonna take that. So I'm gonna call you Xavier only. Okay, you are a research officer enrolled in NIT Calicut PhD program, a school in biotechnology. Okay, nice. I have just one question. You are already using a doctor in your name, right? And you are enrolled in PhD. So are you practicing medical doctor also? Are you practicing?
Yes, madam. Actually, I'm a postgraduate in homeopathy. After
Oh,
BHMS, I did my MD in
okay.
homeopathy. And thereafter, I'm actually already working with central government, with Ministry of Ayush.
Okay, very nice.
And like obviously, we need research work, collaborative research work, and that's why like I went ahead with my PhD program
Okay.
with NIT Calicut. like
Yeah,
that. Just for a collaborative, I'll get more, knowledge in working with collaborative studies with other schools. And
correct.
this time NIT was taking Ayush graduates, and that's why.
Oh, very nice. So yeah, since you are unmuted, what's your first name? How it is pronounced?
Different.
Your first name, how it is pronounced?
Cynics.
Phoenix. Phoenix, right?
Yes, yes.
It's a very unique name. What it
Yes,
means?
yes. It's actually taken from the word scenic,
Okay.
beauty, like that, no?
Yeah.
Natural, like that.
Okay,
Yeah,
you are, I guess, the first person in my lifetime whose name
that was
starts
my
with X.
father's.
Very
Yeah,
nice.
yeah. No, but there are 7 people with my name. I've already
Oh,
researched all over the world, 7 people. There is one IT firm with my name. Yeah.
very nice, very nice. Very like
Yeah. I did a small
respect
research
to
on
you
it. Yeah.
parents who have put such effort in naming you.
Very unique. Oh my
Okay,
God. Yeah. My
imagine.
father's discovery.
Great, great.
Yeah.
So, hi Pujita.
Yes.
Okay. Yeah, kindly mute yourself. Huh? Yeah. So, hi Kanithi. Um, from what course you are enrolled in your university and which year? Please, basic information, guys. Okay, you are from Muthul. Zafira, you are from.
Good evening.
Hi, who's this? I cannot
I'm
see.
Pujita, ma'am.
Okay, I guess there's some network issue maybe from my side or your side, but your voice is breaking. Can you repeat, please? Hello? Okay, it's okay, no problem. We'll talk in between. Hi, Jayashri. I know you have joined NPTEL Animal Physiology course, but what you are pursuing in your uh, career right now? Can you tell me that? Hi, Sayed. Uh, yeah, biomedical engineering. From where you are doing biomedical engineering, Ibrahim? Graduation, life science from Presidency. Okay, okay, great. Hi, Rakshana. Zoology. Okay, similar colleges. Hi, Vishwa. Oh, many biomedical engineer students are joining my course. How come is this your like prerequisite or you guys are crediting it or something like this? All the biomedical engineers drop a yes or no in the chat box if you guys are crediting the course for your like fulfillment of the credit in your college also. Then hi Puksha, I hope I am pronouncing your name right. I'm very bad in like remembering and calling names. I'm sorry for that. Like look at my name, it's so easy, right? Himanshi. My parents didn't put much effort in naming me. Hi Priyanka. Yeah. Okay, great. Priyanka, from where are you doing and what uh you are doing? Kindly drop a message. Hi Gayatri. Okay, there are many PSGR college people. Very nice. Oh, assistant professor in biochemistry. Pungothai. I hope I am pronouncing it right. If you are comfortable, kindly unmute, and I am intrigued that you are also a doctor and an assistant professor already. So I want to know why you have joined this course. If you are comfortable, kindly unmute. Uh, Dr. Anna Durai. Yeah. And if you're not, then no problem.
Good evening, ma'am. Myself, Dr. A. Pungade from Biochemistry Department. I am working in Sacred Arts College, Tirupattur. I am already course completed in NPTEL Industrial Biotechnology, Introductory Cell Biology. So I am eagerly interested for that. So that's why I choose this course in animal physiology. I am handling animal physiology in UG level, but I am also include. I interested if this course will be completed in my life in career. So that's why. And one more thing. methodology also registered for that.
Research
It
methodology.
is it is nice of you that NPTEL courses all things, the assessment, assignment and everything, ma'am. Really very good. Thank you so much for your given this much of scores in Swayam NPTEL.
Yeah. See, it's not at all. My only input for NPTEL is to take live session and help you guys out for this particular course. NPTEL is a really good initiative by the GOI. Many students have told me, and also NPTEL team in personal, that they have earned a huge amount of knowledge and information, and it actually helped in their career. So over and all, it's a good initiative and a good overall project, I will say. So I'll convey your regards to the NPTEL team for sure. You can also do the same on any social media if you are comfortable with it. Thank you so much. Then
Ma'am,
good evening, ma'am.
one more sorry for the NPTEL
Anudurai,
college.
your voice is breaking. If you have any query, you can just drop in the chat box because I'm not able to hear it properly. So I'll reply.
Sure, madam. Okay, sure.
And also other people, if you are unmuted, kindly check your mics. Do mute yourself because it creates disturbance, right?
Excuse me, ma'am.
Yeah, who's this?
Ma'am, I'm Mansi from Haryana.
Yeah, hi, Mansi.
Ma'am, as you are already doing a PhD in from IIT Kanpur, I have a personal question from you that where do you see biotech department in the next 8 to 10 years, as many people discourage me not to take biotech as it doesn't has many jobs or scope in India. So I need your view on that.
Okay. Okay, it's a very heavy question at a very beginning of this course. So if it is possible, can we discuss this question at the end of the session, right? Because we have certain time restrictions. So after we'll go through all the questions, I'll definitely have a discussion with you. Is it okay with you? Is it fine? I'll surely answer that. Don't worry about it.
Sure. Yes, ma'am.
Yeah, don't worry about it.
Good evening, ma'am. My
yeah.
name is Gauri. So I actually had a doubt. So when you say that now it is an online course or online lecture, and there are like 5 to 6 some lectures in whole week. So we have to take them also online, like live, or there are recordings.
They are recorded lectures. You can download those videos, you can download the notes, you can download the transcripts. It is completely user dependent how you want to take your course. These live sessions, whenever you receive a mail for a live session, it will be for discussing previous year questions. And what are these questions? Every week has 5 to 6 videos, right? I hope you guys know where to find the videos, right? If no, just drop in the chat box, and at the end of the session, I'll show you how to screen through those videos. Okay. So there are pre-recorded lectures for this course, right? And it's a 12-week course, which means there will be 12-week sessions in your profile. But every week, one session is activated for the user, which means this is your first week. So first week lectures have already been activated for you guys to go through them, correct? Even second week lectures are live. I guess you guys might be receiving regular mails whenever the week sessions are activated. Okay. So in first week, you will have 5 to 6 videos, which are not live. They are pre-recorded. At the end of that week, there is an assignment associated with every week. Okay. So those lectures you can take however you want to take. After that, every week, particularly for this course, we have 3 TAs, as I've already mentioned at the beginning of the session, right? So those 3 TAs, we will take separately 2 hours live session. Live session means we're gonna, whatever you have learned throughout your week, right? Because you guys are following the video, there might be certain problems or issues or doubts, because you cannot directly approach professor, right? Because those are pre-recorded ones. So how you can clear your doubts? You can clear your doubts by attending live sessions. And there will be 3 live sessions for this particular course. So you can attend any session according to your convenience and ask your doubts. And also we will be solving previous year assignment questions, which might help you in solving your current week assignment questions. Am I clear?
Okay, ma'am. Okay, thank you so much.
Yeah, no problem. Hi, Hari. Okay, great. Yeah, Narendra, what you are saying? See, I'll show you, but I will, as Narendra, I guess you haven't gone through the lectures at all. So I would suggest you to first explore it yourself. In the NPTEL or Swayam portal, where you have registered for your course, the videos are there. Whenever you play a video, there is a link associated with the YouTube app, right? You can go to the YouTube app and you can download like a regular video you download for YouTube. And if you do not know how to download videos from YouTube, there are YouTuber downloaders where you just need to copy paste the link of the video and it will be downloaded automatically. Okay. I guess we should start our session, and we are already 20 minutes late to it, but it's okay. I like chatting with you guys. So I hope I've cleared most of your doubts. There is one question which is there for me to handle at the end, which is where I expect biotechnology in next 10 years. A very heavy question, Mansi, but okay, we'll go through it. Done. So I hope you guys, most of you guys have already gone through the first week lecture. So anyone can brief me at least even a 1 word, 2 word, 1 sentence, 1 paragraph, whatever. What basically you understood from the first week lecture, or what you guys have covered in the first week lecture. If anyone wants to volunteer, just raise your hands, because we all are in a classroom, so we need to follow this, irrespective of whether you are a practitioner doctor or a PhD graduate. I'm sorry, but we have to follow this. So anyone remembers what you guys have studied in the first week of animal physiology? Anyone? And none of you asking about my introduction. No one is interested. It's okay, I won't tell you.
Ma'am, I asked about your masters. You haven't answered it.
I know. Okay, so before starting the session, hello everyone. If you do not know till now, my name is Himanshi and I'm a third year PhD student, PMRF PhD candidate in IIT Kanpur BSB department. I did my MTech in biotechnology from Anna University, and I did my BTech in food technology from Amity University, right? And a fun fact about me is that as I am very easily bored, and you can see in my career that I did BTech in some other field, MTech in some other field, and currently I'm pursuing PhD in a computational biology field. So this kind of a fun fact about me, right? Okay, so now someone needs to volunteer. What we have studied in first week. Okay, just tell me what is animal physiology. Let's start with this question. What is animal physiology?
May I? If you...
Yeah, and please from now onwards, just try to raise your hand.
Yeah, sure, ma'am. Sure.
Yeah.
Ma'am, so animal physiology is the study of different systems like nervous system, circulatory system, digestive system, and how the functions of these systems are integrated with each other. The study of this is known as animal physiology.
Okay. Great, it's a very neat and clean definition for this. But why we need to study animal physiology? This question is for everyone, not specifically for Mansi. Anyone can answer this, because you guys are attending this course, right? From your hectic schedule, you have taken out some amount of time to study animal physiology. Yes, Xavier, go ahead. Unmute yourself.
Yes, madam. Actually, when we do this graduation, especially in BHMS and any medical field, the first year itself, we are studying the normal anatomy and physiology. Okay, so basically, when we understand what is normal, that's when we can understand what is abnormal. So physiology is normal only. That is, taught in the first year, and then from second year onwards, we move on to pathology. Yeah. So I think for us to understand anything abnormal, we have to know what is normal first. So animal physiology, I attended the first week lecture. It's talking about like mostly says very similar to physiology that we are taught. So I find it very relatable. But then also with every animal, like sir's trying to correlate with the lower animals also, apart from only human beings. It's like a general lecture. So that's why I think knowledge of animal physiology is important to know what is abnormal before uh, like for that we need to know what is normal.
Yeah.
Yeah, thank you.
Correct. So in a crux, can we say this, that see, we are self-declared the most important species on this planet, right? We are the humans. We are most concerned about our living, right? No other animal is, they are concerned, they survive, they thrive, right? But none of them are conscious enough to work only for themselves. We are the only species who look into a factor of living long and also the quality of life, right? No other animal is bothered about quality of life. They are only bothered about living, right? So to understand or to enhance the quality of our life, there are n number of sufferings, but those sufferings can only be understood, as Dr. Xavier clearly mentioned, right? We can only understand abnormal things when we know what is normal, right? Because if something is black and the other thing which is not black, then only we can say that it is white and it is not black. How come you will know that this is white? Because firstly you need to know that, okay, this is what the characteristics of a black color is. These characteristics are completely opposite for a white thing, right? So one thing has certain characteristics, other thing has certain other characteristics, then we can make comparisons, right? So this is one of the basic way of how we can enhance or understand also our living, our say way of doing things also. If something make us say struggle a bit, for example, if the environment is not comfortable for us, there is too much humidity, too much heat, too much cold, right? We get bothered about it. We always seek for the optimum level. And it is not only we, all living organisms, right? And even a virus particle, which is neither living nor, sorry, not living, neither your dead particle, right? So even when it gets the suitable environment, it replicates really quickly. Talking about living organisms, we always seek for the optimum in everything. As in the lectures, those who have went through the lecture, there are certain major properties or major macro system properties which we are more attracted towards, and we want specifically those criteria to be optimum for us. One thing can be what? Temperature, right? As most of you, like if I remember correctly, you are from Tamil Nadu, right? Towards from the south. So the heat is too much, the humidity is too much. But the moment if you migrate towards extreme north, say Jammu or Kashmir, right? Your problems will be sorted. At that time, what was your problem? Your problem was it is too humid, there's too much heat, and like it's too hot there. So what was your problem? These two factors. So you cancelled it by moving ekdam poles apart from your state, right? But again, you won't be comfortable there. Why? Because there's too much cold. There is too much dry air going there. You are feeling dry. Your skin is dry. Like your thirst is not quenched, right? So we always look for the mediocre thing. How we can do this? Okay, there's too much hot outside, we'll put AC for us, right? If there's too much cold outside, we'll wear all the good wool clothes or quilts, we'll use everything, room heaters and blah blah blah, all those things. So our system wants to be in the optimum. And why we like optimum? Because all the physiology from, there were levels, right? If you remember in the discussion of first week lectures, the chemical or molecular level, right? The cellular level, from cells, the tissue level, from tissue level to organ level, from organ level to organ system level to we, the organisms. So if our small molecules or chemical molecules or biomolecules are not happy in particular temperature, pH, oxygen content, pressure, or say even stress, right? Then if our smallest unit is not comfortable, how come whole organism will be comfortable, right? It's a polymer. You get my point? If you have proper balance of small molecules or biomolecules, your whole, indirectly we can say, because obviously there's an increase in complexity from biomolecule to organism. But in a very simple and straight way, can we say this, that if our small molecules or biomolecules such as our proteins, lipids, carbohydrates, right, and our nucleic acids, they all are in their best condition, an organism can be in its best condition, right? Though it is not directly related, I'm not saying that, because there are, as soon as you're moving from small molecule to cell to tissue to organ system, the level of complexity is increasing. But to simplify things, we can assume this, right? So this was the whole sole purpose for you guys, specifically in the first week of lecture, to understand first thing why we need to learn animal physiology, correct? Because in the subsequent weeks, you will be dedicating your time in understanding one system at a time, right? So first week was completely like a overview of what to expect, why we need to study it, how to study it, and how to look for different things. Am I clear? Anyone has any doubt in this? Yeah, Samadrito, I'm reading your message now. It's quite a long message. Since birth, we have changes which are irreversible. Our daily life can be viewed in perspective of physiology. Asking why it happens to our body is related to animal physiology, correct? Our life loves stability, really nice. And our body loves homeostasis, great. I didn't wanted to use this term, but okay, if you have brought it up, we'll discuss it. And to maintain homeostasis, there are certain sensors, hormones, and neural control. Great, you have like almost summarized the first week. Good job. So I guess you have answered for the question when I asked for the first week now. Great. So I guess with this very brief culmination of first week things, we can start with certain demo questions. And don't worry if you don't know the answers, but I believe most of you guys will know the answer because these are very basic questions. As it is first week, the questions will get difficult, like tougher and tougher as we progress throughout the course. So if you know the answer, type in the chat box. If you don't know the answer, don't be discouraged. We are here to help you out. Okay, great. So we'll start with our first question. Okay. So our first question is, in anatomical terms, the blank space plane is a vertical plane that divides body into left and right portions. So if you know the answer, I'll give you guys 1 minute. Just drop your answers in the chat box. Okay, I have already received few answers. Great. I need minimum 50% participation. Otherwise, you guys I will make it mandatory for you guys to like turn your videos on. You need to participate at least in the quizzes. Okay, we have few answers. Till now, we have competition in A and B option. More supporters for A and B, or C or D even. Come in front. Great. Keep coming, keep coming. And don't need to be sorry. It's okay. You can change your answer. This is not your exam. Completely fine. Okay. Your 1 minute is about to get over. So everyone just write down your answers. Okay, great. Oh, I'm happy that you guys all are participating. Nice. Okay. I guess most of you guys are, what do you think? Correct or wrong? Okay. So most of you guys are correct. It is called sagittal, right? And to understand this in somewhat better way, you can always remember this. It's a very representative image of all the ways we can divide a vertical plane, right? And anyone can tell me why we need to divide particular plane? Why can we it into these many number of ways? Any hunch, any guess?
To see the proper working of organs on the both
Please
sides.
always raise hand, please. Yeah, go ahead.
I'm sorry, ma'am. So
It's
I
okay.
was saying, we need to know the proper working or functioning of the bodies on both sides.
On both sides. But
Like
we
for example, if we have like left and right lobes of our brain, okay, so they have both different functions. Like left for like that. So I don't know the specific, but I guess like we need to know the perfect functioning or the we need to know the all of their
okay,
functioning.
okay,
Like
no problem. I appreciate you for your uh guess. You have a bit understanding of it, but it's not exactly what it is. And we'll give, okay, we have few more participants. Uh, Xavier, I'm gonna ask try to Mansi. Yes, Mansi, go ahead.
I think we divide the body in a particular section to have a better 3D anatomical view of the body, so that we can, use it in the medical field to do surgeries and to have um, to perform surgeries. Yeah.
Hmm. Yeah, good job. I guess this sums up the thing. The whole zest of it is in it. And so, yeah, Xavier, do you want to add something in it now?
Madam, basically the sections are done to understand study these sections when we are studying the anatomy. Other terms used when we are studying the anatomy of the brain. So it is more to do with the structure. Where is which side? Left-sided. Sagittal is like the front. It's like you can just draw how I used to remember and how I could remember it so fast. Is remember the word S, okay, and draw it, and it is from the front side, okay, dividing your head into right and left. So that way.
Great
So it
image.
is more to do with the structure and how you divide the anatomy of the scalp rather than anything to do with the physiology.
Yeah,
Like
yeah,
that. To
yeah.
know the location, exact location of your particular part of the brain or like that.
Yeah. So overall, this is what I guess all of you have culminated into it, that we need to understand the structure. See, if we are studying something, right? For example, let's take anything. I guess if we take the structure of this phone, right? And I want to study it, correct? If you want to study it from a surface level, what you can study? You can just study this first part, the screen of it and the back of the phone, right? But the actual functioning of it is not happening on the screen, correct? It is happening in between the screen and the back side of the phone. There are circuits inside this. And if we do not know their position or structure or where they are located, we won't be able to do anything about it. So first, to understand where is our structure present, to visualize it, to see it, then only we'll be able to do something about it. Whether, and that this doing, it can be anything. Either it can be learning about it or performing surgery to it or just observing it, right? It can be any operation. So to do anything, we need to know its proper location and structure. Also, our current techniques which we have for imaging, they have been improved. Gradually, they are improving also. But if you talk about earlier days, the only thing which was present for visualization purposes of a, say, human body or a living 3D structure was X-ray. Even now, X-rays are available, correct? But X-rays won't give you all the information, right? Correct. You won't be able to see soft matter there. You will be able to see bones and like solidified masses, but you won't be able to see soft matter. Then we have like there are other imaging techniques now which are getting better and better to get the 3D information of it. So earlier, when we didn't had any technical advantage towards us that we will be able to visualize 3D, what we came up with, we said, okay, it's a 3D object and we don't have particular technique for it. What if we will try to slice it into smaller particles, and we can study smaller sections, not particles, sorry, and we can study smaller sections, correct? So this is that approach, one of that approach. But as the time grows, our techniques advanced, and I guess now we don't have much issue in imaging 3D objects, but still we would like to know the exact position and location. So we merge all the techniques, like 2D techniques as well as 3D techniques. Done? Okay. We'll move towards the second question. Yeah, so our second question is, which of these muscle types is not, or sorry, it's non-striated? Cardiac muscles, skeletal muscles, smooth muscles, or none of the above? Your 1 minute starts now. Oh, great. Everyone is sending me answers. Nice. Okay. Okay. Oh, we have very inclined answer. The option C is winning. Is there anyone who wants to take a chance and answer something else? Okay. Okay, great. So we can see that our answer C is winning. So I guess we should not take much time in this because this is an obvious question, right? So our correct answer is obviously smooth muscles. Kudos to all of you who have answered correctly and still answering. Great. So I don't think so that we need to put much time in this, but this is just a very basic thing about muscles, because I guess we haven't covered much about muscles in our first week. We will cover it in our subsequent weeks. So this is a very brief information about what are skeletal muscles, the basic features of skeletal muscle, features of smooth muscle, and feature of cardiac muscles, right? Yeah. And don't worry, you will all receive this video, like whatever we are discussing, it will be uploaded on the YouTube. You will receive the link for it and the presentation also. Okay, so don't worry about it. So our next question is, it's very easy one, and I expect all of you to answer it. Oh, let me finish reading it first, guys. Yeah. Dendrites and axon are part of which cell type? Neuron, nephron, mast cell, or glia? Oh, I guess it's a clear winner here also. Okay. I'm so happy that all of you are participating actively. More answers. I've appreciated you guys and you stopped messaging me. Okay. Oh, okay. Okay. I guess all of you have answered it. Really nice. Again, there is no explanation required for it, right? We know what a neuron is. Okay. Quickly tell me, nephrons are associated with which organ?
Kidneys.
Type in the chat box. Yeah. Yeah. Gauri, do you have any doubt? Your hands are up. If yes, kindly unmute yourself and ask. Yeah, correct. It's kidney. Mast cells are associated with which organ? I'm asking about organ. Hmm. Ooh. Sorry, I'm extremely sorry. It is associated with immune system, not immunology. Narendra, immunology means study of immune system, right? And we are asking about to which system or organ it is associated with. So a better way of saying will be immune system, right? And glia is associated with mast cell. Mast cell not associated, but certain mast cells like Kupffer cells are present in liver. Yes, but all these come under immunology, right? Samadrito, correct? Yeah, they come. Their name changes based upon in which organ they are present, but at the end of the day, they all belong to immune system because they are defensive cells, correct? And glia cells is associated, great. Glia cells, neurons are associated with CNS of brain in general. We are not using too technical terms because it is just our first week. So we can go in a very like broader manner. We are not going too deep into specificities. Great. I like you guys. I like all of your enthusiasm. Great. Keep going. Our next question is in integumentary system. Which of these is not part of cutaneous membrane? Is it epidermis, dermis, papillary layer, or nails? Okay, great. You guys have 1 minute for this question. Okay, we are receiving some answers. Oh, you guys already know so much. Great. Okay. Okay. I'm reading all of your comments. I might not respond to every single one, but I am reading all your comments. Okay, great. Very nice. So obviously the answer is nails. And if you don't know, those who are confused, don't worry, we will be covering all this in our subsequent lectures. I know, Samadhito, it is not present in our first week lecture because these questions which I am discussing, these are a bit overall things. After, I guess, after 10th slide, you will get to know that the questions which I am discussing, they actually belong to first week. So don't worry about it, as I have already given you guys disclaimer, right? If you don't know the answer, don't worry about it. We will discuss everything, correct? If you don't know the answer, it means we haven't covered it in our lectures. Don't worry. Right? So what is the integumentary system? It is one of the largest organ of the body that forms a physical barrier between the external environment and the internal environment that it serves to protect and maintain, right? The integumentary system includes epidermis, dermis, hypodermis, glands, hairs, and nails, but nails doesn't belong to cutaneous membrane. Great. Next question. And yeah, Samadhito, don't worry. After 10 slide, you're gonna answer correctly. So our next question is, variations in hair color is because of light around us, food we eat, structural differences, and variations in amount of melanin, or all of the above? Ah. I have a very interesting question for you guys. Whoever is answering C now, I have a very interesting question for you all. Keep answering, keep answering. Don't stop. I might confuse you. This is my job. So be stick to your foundations. I might test them. Yeah. Okay. We have received, I guess, more than 50% of the participants. Still, 1 minute is my job for you guys so that you can clearly read the question, understand the question, then answer it. Keep them coming. And also read every option one more time. Yeah. So the answer is, it is C, obviously. Why not? A little bit of extra information you can read at below. But my question to you guys is, correct. This is where I was pointing to Samadhito. How can you guys completely eliminate the option A? Anyone? Those who want to answer, just raise your hand. If you want to take a guess also, don't worry about it. Not necessary, Mansi, not necessary. Melanin is just a pigment, right? Its expression level differs in different parts, but it is associated with colors. And also you can look at the bottom information, right? There are different types of melanin which are associated with different colors. Okay. So your answer is absolutely correct. Wherever you hear that the hair color is due to, the answer will be pigment associated. It won't be any external factor. But I want you guys to think why we are completely eliminating the effect of external factors. You have heard that, right? Uh, Due to excess exposure of UV or sunlight, the graying of hair increases, right? It accelerates, correct? Because I am suffering from it. So yeah, it's a very generic thing. Even nowadays, like small kids are having gray hairs, though they have this pigment, correct? But their hairs are turning into gray. One reason is the sunlight also, because of the damage and blah blah blah, other like different things associated with it. Food we eat. So agreed, we cannot directly say that if we eat dark colored food, we'll be having dark hair colors. Completely agreeing with this. But don't you think nutrition has some role in our hair growth, right? Like all these advertisements around supplements, especially biotin, right? Supplements, hair oils, shampoos, and they all revolving around strengthening your hair, increasing the hair growth, right? So directly or indirectly, we can say that the food which we are having might affect the growth of our hair, correct? So I'm not saying that your answer is wrong, but I'm just saying that they are not the direct factors which are associated for the color of your hair. They are associated for the things which comes after coloring. Since birth, if I guess you guys will be pretty much aware about the inheritance, right? Certain traits which are inherited, certain traits which are adapted. So the traits which we are receiving from our parents, right? They determine the color of our hair. And throughout the life, whatever changes are happening, those are adaptive things, correct? So light around us do affect our hair, but they are adaptive. They are external factors. They are not determining our hair color, but they are changing the quality, the quantity, as well as the texture of your hair. Similarly with the food. So in future, if you encounter such a similar question, remember to read the question very clearly, because here we are directly asking variation in hair color is because of. Here the variation is not considered for the things which happen after birth. Variation is accumulated during birth itself, right? So these are the variations. So our hair color is determined by our parental genes. They are not determined by our external factors. Correct? I hope it makes sense. We'll move to next. Yes, Mansi, they are temporary. Correct. So what is keloid? It's a very generic term. If you know, it's fine. If you don't, it's a very basic term. So keloid is a bone tissue, hair tissue, lung tissue, or scar tissue. Your 1 minute starts. Oh, you guys don't even need 1 minute now. Great. Okay. More answers, guys. Your frequency is reducing. Start. Great. Was this discussed in this week lecture? If yes, give a thumbs up. If no, don't do anything. Samajito, don't worry. It's okay. We'll get to the questions for this week specifically. Don't worry. And I'll ask you only. No, I'm not going to do that. Okay. So we have answered. It's a very generic term. It was not discussed in this week, but it's a very generic term for scar tissue. And if you don't know what scar tissue is, what's or specifically keloid is, I guess some of us have it, right? And or we know someone who has these scars. These scars are developed when a particular wound is not healed properly. There are different stages of wound healing, right? When you injure yourself, the first thing what happens is, anyone of you wanna give it a try? Except the practicing doctors. You guys are my last resort. So anyone wanna give it a try? Like, if you enjoy yourself, like I guess many of you, and I guess every one of you at some stage of your life might gotten some injuries, correct? So the moment you enjoy yourself, what is the first thing that happens? Anyone wanna give it a try? Raise your hands. It's okay if it is technically wrong. Don't worry about it. Don't worry about the terminologies also. Don't. It's a very common thing. What you observed when you got injuries. None of you have gotten any injury. Seriously, it's like good for you guys. Okay, all the doctors in the house, the question goes to you. So what happens when you enjoy? What is the first event that occurs when you enjoy yourself? Anyone? Raise your hand. None? Yeah, please go ahead, Xavier.
Yeah. So
Sorry?
first, whenever there's an injury, any kind of injury, first is vasodilation. There's a rush of blood to that particular part. That will be followed by the body's mechanism to stop that bleeding, which has got vasoconstriction and then platelet fibrin aggregation. There is basically scar tissue is of two types. One is hypertrophic scar and one is keloid. So this definitely only a medico can tell. And even the picture that is shown is a hypertrophic scar. And for keloid, the only most common location is the chest. You must have seen someone had a small boil and they burst it up on the chest, and then it kept on the person kept on scratching, and it grew into a huge. It's a bit larger on the chest region, no? And it keeps on increasing. Even if you remove with surgery, it will come again. So that's an abnormal wound healing, which we will call a hypertrophic scar or keloid.
Great.
It could be either way.
Thank you so much for this information. Everyone, don't worry about this. You all will know all these terminologies. Don't get scared of this. So common location of keloids is obviously not this. This is what this is like an injury which was not healed properly. Keloid is somewhat developed, like as Xavier mentioned, in chest, ear, cheeks, and all those places. But basically a scar tissue, right? A scar tissue develops whenever there is improper wound healing, right? There are, I guess, 5 to 6 steps for a wound to be healed, right? Because the moment you injure your body, like your whole system is working only towards healing that system, right? Because your primary and most strong barrier from the outside world is your skin, and we have a rupture in that barrier, correct? So our system is such like fascinating system, and it is so much integrated that as soon as one barrier is like impeached, what will happen, our whole system will try to close that barrier as soon as possible. So during this process, what happens sometimes is that due to external factors or due to like improper signaling at your wound site, it doesn't happen as it was expected to be. That is why some people, for example, there's a like, even if we are not talking about scar tissue, let's take blemishes. I guess everyone will relate to it. Like you get pimples. So for some people, the pimple gets clear very easily. Like within a week, there won't be a single spot, right? For others, it might take more time. So it depends from person to person and their biological needs, how they are like daily lifestyle, how they are spending their time, are they treating it, are they doing anything for it, or they are not doing anything for it, are their nutrition enough or not, correct? So whenever there is imbalance in any stage of wound healing, a scar tissue, a spot, or a inflammation is the first step of wound healing and scar development in most of the things, right? So this will happen. Don't worry if you are not understanding most of it. It's just a side information. It is not at all related to keloid or your first week lecture, so don't worry about it. We will discuss all these things in coming week lectures. And also, if you are not discussing it, you guys can always post question and I will also learn from you and I'll get back to you, right, if I don't know any answer. Yeah. So our next question is, C cells of thyroid glands secrete what? Is it calcitonin, calcitriol, thyroxine, or growth hormone? You guys have 1 minute. Please answer. Okay. Cool. I guess we have received enough. You guys still have 30 seconds to go. So quickly drop in your answers. Yeah, I guess most of you, all of you are correct. Oh, you guys should teach me. Yeah. So obviously our correct answer is calcitonin. C cells secrete calcitonin. So why I'm showing this image? Because except all the trained doctors and scholars here, most of you guys might are still in your first year or second year, right? So you might not have seen real cells, how they look. In books, most of the time they are like represented. They are not shown as a real cell. So I want you guys to get comfortable with imagining cells as how they actually look, correct? Because in a representative way, we just draw a circle or an oval, correct? But in reality, they are not like that. So the reason behind putting this particular image, the cells are there for you to understand that how they actually look. Okay. So get okay with this thing. And whenever we say cell, try to force your mind in picturing the real image of a cell. For example, if I'm saying, say nephron or neuron, We have the representative image of it, right? How we have drawn it in the books. We overly simplify things in textbooks so that at least you can get the concepts right. Because the first thing, it should be the concept, correct? But always go back to the net. We all have access to internet, right? Always go back to the net and search for the microscopic images of those cells, that how they actually look instead of how they have been represented throughout the books or papers or journals, right? This will help you in understanding the beauty of our system, as well as you will understand the complexity which are associated with it. Okay, so this is just some stimulation for your imagination, correct? So all of you must do this. It will overly simplify your understanding, and it will help you in relating your understanding to the real life scenarios also. Okay, our next question is, the dash is a ring like structure containing melanocytes and is part of the eye. So is it ciliary muscle, retina, iris, or cornea? You guys have 1 minute and your time starts now. Everyone drop your answers. Yeah, great going. You guys have 20 seconds to go. Everyone just put your answers. Don't be scared if it is right, wrong. Don't worry. It's an open platform. Even if you are wrong, don't worry about it. We all are here to learn. Don't worry. Okay. Okay, your time's up. So obviously, it's pretty simple, right? It is iris. And I don't think so we need to discuss more. And in the coming week, I guess it will be third or fourth week when we're going to discuss about eye and eye structures. So we'll go in depth into it. Cool. So now I have a question for Samadrito, right? Are you there, Samadrito? Give a thumbs up. Oh, you're not there. Don't worry, I'm not going to ask you any question. Just give a hands up if you are. Okay, great. So now tell me, was this question a part of this week? The plasma membrane is a sulfated monolayer, phospholipid bilayer, rigid structure, or red colored structure? Oh, everyone's answer is coming. Guys, wait, let me see. I have to give you 1 minute time. Think, read. At least give me some complex answers. Yeah. Okay, keep coming, keep coming. Great. All of you guys already know so much. Okay. You guys still have 17 seconds. Those who have not registered your answer, it's your time. Start. It's very... Yeah. Okay. Yeah, your time's up. Obviously, the correct answer is phospholipid bilayer. But I have one question for you guys. As you have very quickly answered this particular question, why not a plasma membrane can be sulfated monolayer? And I know you guys might not know about it, right? But there's a particular reason, right? Why sulfated monolayer is not there. So even if you don't know the exact answer, even I don't know the exact answer, but I want you guys to start thinking in that direction, that why we cannot have a sulfated monolayer. Yes, Mansis, go ahead.
Ma'am, there are some embedded proteins which are embedded in the phospholipid bilayer, which helps some material to cross the plasma membrane. So it is a lipid bilayer, phospholipid bilayer.
But it can be sulfated also, right? Why not sulfated? Like, what is wrong with sulfur? Sulfated monolayer means it's a, okay, leave the part of monolayer, right? And my question is not only to you, Mansi, it's to everyone out there. Leave the monolayer part. Okay, we are not, because we know that plasma membrane is a bilayer, so you can easily exclude the first option. But I'm saying, what if we have a sulfated bilayer? What is wrong with that? Yes, Gauri, go ahead.
Ma'am, as phospholipid bilayers are actually hydrophobic and hydrophilic, which actually gives a stable rigidity to the membranes. And also they are a good transporter, like they are very able to transport because of their permeability in them. I guess that's maybe the answer.
Okay, so you are saying that phospholipid bilayer are hydrophobic in nature, which is correct. And then you are saying that it helps in transporting some XYZ compounds across it, correct? Like, did I follow you right?
Yes. Also, it gives a rigidity and stability to the structures. They are very stable for cell membranes to be made of.
Okay. See, here what I want you guys, see, whatever you guys are answering, it's absolutely correct, because since the beginning, what we have read, that our plasma membrane is lipidic in nature, proteins are present, it's bilayer, that bilayer is made up of phospholipids. It's absolutely correct. But what if we didn't have this discovery, that we don't know that it's a phospholipid nature and it's of bilayer in nature? So sulfated means, I guess you guys are not able to understand what sulfated means. So take any compound, put sulfur to it, right? Because in this option, we haven't mentioned whether sulfated monolayer of what? Is it a protein which is sulfated, sorry, or is it a lipid which is sulfated, or is it a nucleic acid which is sulfated, right? Sulfur is a small molecule, correct? Sulfated means something which has sulfur. So in this option, nothing is given. So I want you guys now, for example, if I say there's a, let's take X compound, and X compound has sulfur. Those X compounds are like packed together to form a bilayer, right? So you have XXX compound, and every X compound has sulfur to it, correct? And they're stacked together just like a bilayer. So is it a possibility, or it's a complete myth? Not myth, I mean, or is it a complete fiction? Can this possibility occur? Imagine you don't know that layer is made up of phospholipid. Can form, Samadrito is saying that the sulfate, I guess he's suggesting for sulfated layer only, key, they can form disulfide bonds causing rupture of cell. Samadrito, if you're comfortable, you can unmute yourself, but I have a question for this, that if something is making a bond, like disulfide bond, it's a covalent type of bond, if I'm correct, right? So if something is making bond, how can it rupture the cell? Because you're making two things bonded together, you're making it stronger. So how can it rupture? So I just want some clarification on it.
No, ma'am. Actually, if the sulfur is suppose present at position number 1 and another sulfur is present at position number 100, if number 1 sulfur and number 100 sulfur makes a covalent bond, it will cause rupture. Like cell will squeeze out.
Okay. So here what we need to do, see, I'm very bad in drawing, but okay, I'll try for you guys.
Ma'am, may you please repeat the question? It is not clear to me.
So I was asking Samadrito, if you look in the chat box, he's saying that if sulfur is present, right, in a bilayer or a monolayer or in a plasma membrane, if sulfur is present, that sulfur will make disulfide bond, as a consequence of which the cell will rupture. This is the point which we are discussing right now. Are you clear with this point?
Yeah, ma'am. Yeah.
Okay, great. So, Samadrito, what you suggested is that if we have first sulfur here, then we have something, 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 and this is the 100th sulfur, right? This is first, this is 100th. And I have already said that I'm bad at drawing, so don't judge me on this. Yeah. So you are saying that this first sulfur will make disulfide bond, which is a covalent bond, with 100th sulfur. Correct? Am I correct till here? What you are saying?
Yes, ma'am. Yes.
Yeah. So don't you think that, I guess you might have studied in chemistry, that there's a particular range for a covalent bond, right?
No, actually, I want to mean that if somehow 100th sulfur comes near to the position number 1, or it might be possible that another cell sulfur binding with another cell sulfur.
Correct, correct. So I wanted to bring your attention to this point only. This is 1D or max to max 2D representation of thing. But in 3D representation, what will happen? This is your first sulfur, right? So suppose it is in, this is your x-axis. I'm sorry, from next class onward, I will use pen, okay? This is x-axis, this is y-axis, and this is your z-axis. So if one sulfur is present in XY plane here, right? Another sulfur is present in, maybe this is the first sulfur. Your next sulfur is present in YZ plane. Are you guys able to imagine the XY plane and the YZ plane? Just, I don't have a notebook here. Just take a notebook, right? Take a notebook. Just imagine this is my notebook. This is x-axis, this is y-axis, and this is z-axis. So if something is occurring in this plane, other thing is occurring on this plane, so they are technically in 3D position near to each other, right? Are you guys able to imagine? I don't have a notebook. Just take an example of a notebook. Just put all this x, y direction on a notebook. Correct. So if something is present on XY plane, something in 3D location is present on YZ plane. So in 3D location, they are close, right? Agreed? Yes or no? Maybe anything response? Okay, great. So then they are in the vicinity, in the 3D position, in the 3D location, in the 3D distribution of molecules. Then there is a possibility that if they are closer to each other to a certain distance, they might form disulfide bond. Correct? And disulfide bond are majorly associated with proteins. And here we are only talking about membrane. But let's, okay, if we are fictioning everything, we will fiction it also. Correct? So they are in closer proximity to each other. Chances is this first lipid is lipid number 1, the sulfated one. This is the 100th one. And they'll make disulfide bond as said by Samadrito. Correct? But now my question to you is, if first one is here, and then it can only make a disulfide bond when something is in its close proximity, say here. So this interaction is making my system stable. This is not making it unstable that the cell will rupture. Say, for example, if here, I guess you're saying that if this point is very strongly bound, this will be like, it will put much pressure here and the rupturing will occur here. If I'm getting your point correct, maybe. Otherwise, you can correct me. But always remember this is a very basic concept. Whenever there is bond formation, right, specifically covalent type of bond formation, the system will always get stabilized. How many of you agree with this? Just give a thumbs up. If you don't agree with this, don't do anything. I will try to explain it in a better way. My statement is, whenever there is bond formation, especially covalent type of bond formation, the system
So,
will always
Actually,
get stabilized. Yeah, Samadhito.
actually, when sulfur is present in S form, not S2 form, it always tries to form S2 form. And when S become S2, that will call the rupture.
See, maybe I'm not able to get your point, but I guess I'll definitely come back to you next week. But try to understand what I am saying. Okay, have you observed this phenomena or principle anywhere? If you can give me reference, or you are
Like
just
it's a group 16th element. Oxygen is present at O2, sulfur is present at S2. No, only S can be S cannot be there, or it must have formed any types of bond between themselves. It's more preferable, and sulfur has less negative hydrogen enthalpy. ठीक है, तो इसलिए I guess वो bond कर सकता है.
you are correct here. This is the thing, Samadhito. You are talking purely on the basis of small molecules. Correct. This is what happens in chemical reaction when you write it. But also remember that S wants to be in S2. Why? Because it is more stable in S2 form, right? Then only it is. Every living system always wants to be at its lowest or higher energy level. Everyone write in the chat
Lower
box.
energy level.
Oh, okay. Yeah. Lower energy level, right? Whenever a bond is formed, any type of bond, I'm not talking about disulfide bond. Okay. Whenever any type of bond is formed, the energy is released or is used. And if you're not clear,
Bonding enthalpy release होता है.
तो सिस्टम का एनर्जी बढ़ रहा है या घट रहा है?
सिस्टम का
सिस्टम
एनर्जी घट रहा है.
यानी कि स्टेबलाइजेशन की तरफ जा रहा है. करेक्ट?
Yes, ma'am.
So your statement saying if a disulfide bond is formed, meaning energy is released, meaning system energy is getting lowered, meaning the system is achieving stabilization. If the system is stable, will the system burst or will it try?
System is getting stabilized when disulfide is formed.
Correct.
That means when two cells are present near to each other, the disulfide bond is forming because they wants to lower the system energy. So when disulfide bond is formed, इस system का energy तो low हो गया, लेकिन cell तो rupture ही हो जाएगा ना.
देखिए, okay, are you comfortable in Hindi or English?
कोई दिक्कत नहीं.
दिक्कत नहीं है, ठीक है. यहाँ पे आप दो लेवल को मिला रहे हो, ठीक है. I guess how many of you are not okay with Hindi? Can you just type no in the chat box? Because I want everyone to understand this concept. Okay, 1, 2, 3. Okay, don't worry. I'll explain in English only. Don't worry. It's okay. So, okay. First, you are saying, and this is the first level. The first level was biomolecules or chemical compounds, right? So if two sulfur are present, they will make disulfide bond. This is clear to everyone and this is correct. This is first level of organization, cell and molecular level, molecular level, right? Immediately you are jumping to cellular level. When you are saying, if this sulfur of first cell binds with this sulfur of second cell, the rupturing will occur. Here, the proper term, if you are using cell to cell, it would be like mingling, not rupturing. Rupturing means bursting, right? छूट जाना. तो अगर दो सेल मिल रहा है तो वो टूटेगा नहीं. They will merge into each other. And again, this principle will vary of what thing we are talking about. Are we talking about mating when two cells fission into each other? Are we talking about are we talking about cell division when cell divides into two? Or are we talking about immune cells where there is recognition of like paratope, epitope? And don't worry if you do not know all these terms, we will study all this, right? But I'm talking to those who are a little bit aware about these terms. So if we talk about proteins, right? In proteins, the major stabilization factor for the 3D structure of protein are van der Waals interaction, hydrogen bonding, and also disulfide bonds. correct? So if first monomer of a protein is getting attached to the second monomer of a protein via disulfide bond, they are not rupturing, right? They are getting more stabilized. This way they can work as a dimer now. They are not only not one unit is working, they are working together, making system more compatible with the surrounding, making system more stable. There is no rupturing because the bond is formed here. There is connection, right? There is no bond breaking or rupturing, correct? So the thing which you are saying is disulfide bonds are formed. This is molecular level. Then you are coming to second level of organization, that is cell. And you are saying when disulfide bonds are formed here, so you cannot apply the same principle which works on small molecules to a larger system because their properties might change. Basic principle will remain same. Disulfide bond will be forming here also. Disulfide bond will be forming here also. But here, the molecule, the individual molecules wanted to be stable. Here, the macromolecules or the bigger unit of classification, or say here we are using term levels, wants to be stabilized. There might be certain, for example, if we take the example of porins, right? Porins are certain compounds which make holes in the cell membrane, right? So making a hole, bursting things, breaking things, it all symbolizes breaking of bond, right? Use of energy, increase in the energy of the system. So whenever, so my statement was, and Samadrito, if you are not convinced, it's okay, you can drop me a text and we can discuss it later or maybe next week. But the basic concept which I want you to understand, and Samadrito, if you are still confused, I will ask you to put your two statements, what you are saying, and put it in the light of levels, right? The way I'm trying to explain, at a cellular or small molecule level, and us at a cell level, at a chemical level and at a cell level. So chances are you might understand the discrepancy which I'm trying to highlight here. So to everyone, what I'm saying is, whenever there is bond formation, most of the time, always, because in biology we can never say 100% of the time, no, there will always be exceptions. So most of the time, close to 99%, whenever there is bond formation, it is associated with the stabilization of the energy, stabilization of the system, no breaking of the system. There was a reason that those bonds were formed. This was an individual entity, this was an individual entity, this was not stable in the system, this was not stable in the system. They both came closer together, they made interactions, they made bonds to be both together stable in the system. I hope I didn't complicate it much, and if I did, I'm extremely sorry. If you guys are not clear about it, we can discuss it next week. Okay? To those who have understood at least a little bit of it, can anyone of you just give me a thumbs up or something like this so that we can move ahead from this topic discussion? Okay, I got only 2 thumbs up. Okay, okay, okay. And don't worry if you don't understand this at once. These are some basic things which I'm trying you guys to understand, because everything in physiology, especially, you need to put it through a screening of levels. If you're talking about bond formation, are you talking about the bond formation between two giant entities? Or you're talking about bond formation within the molecules? Or you're trying to understand the bond formation of within a molecule or between small molecules, right? So you need to put them in different filters, correct? Yeah. So, and I'm sorry to those to whom I have confused. I didn't mean to. So again, feel free to ask questions. Even in next week, I'll be happy to discuss. Okay. So obviously the correct answer was phospholipid bilayer, and this is the most detailed representation of a cell membrane. Cell membrane or plasma membrane, it does not only have a phospholipid bilayer, it has many entities associated with the membrane. So by saying that it is sulfated, I meant you guys to imagine that it's a membrane and some sulfurs are attached to it. I wanted to discuss other 2 points also, but due to restriction time, we will move forward. Okay. So, okay. So our next question is, dash refers to the relatively stable. Okay, I've received. Okay, I'm receiving answers. Great. So Samadrito, from now onwards, all the questions are from first week. So I expect you to answer it. Okay. Dash refers to the relatively stable state inside the body of an animal. First, homeostasis, equilibrium, fluid balance, organ symmetry. You guys have 30 seconds to answer this. Quickly, everyone drop your answers in the chat box. Okay, I have received quite few answers for this question. Okay. Yeah, so obviously okay, I'm not gonna tell you. You guys have 10 seconds. Okay, so obviously our correct answer is homeostasis. What is homeostasis? Anyone wanna give it a try? Anyone wanna explain it to me? And I don't want any technical language. If you know technical terminologies, good for you. Go ahead. Don't feel shy in using them. But even if you don't know the technical terminologies, if you know the concept, I'll be happy to listen to you guys. So anyone who wants me to tell what homeostasis is, kindly raise your hand. Okay. I want others also to give try, because there are only few people who are answering. I want everyone to understand. You see, a very minimalistic thing which you guys can do is take maximum advantage of live sessions, right? So if you don't know the answer, don't worry. If you want to give it a try, even if it's a blunder answer that you think ye to ho hi nahi sakta, still give it a try, right? What if you might be right? Correct? And there's no punishment for wrong answers. There's no negative marking or something. You're not getting uh, examined here. We all are here to discuss things, correct? Okay. Acid-base balance that takes place within the body by specific organs. Selena, it's partially correct. This is what I want to say. Do you want to add anything in it? And I guess I'll go with Samadrito this time. Yeah, Samadrito, what do you want to say about homeostasis?
सपोज हम लोग कोई घर में है जिसका टेम्परेचर 25 डिग्री सेंटीग्रेड के अराउंड है, और जब घर से निकलता है तब 46 डिग्री सेंटीग्रेड तो आ ही जाता है ना। तो इसलिए तो दे आर अ सर्टेन एडेप्टेशन वी हैव अडेप्टेड इन द कोर्स ऑफ इवोल्यूशन सो दैट वी कैन मेंटेन आवर बॉडी टू फील इट्स क्वाइट कूल टू cool our body. It may be sweat. There may be occur sweating of the gland, or the water reabsorption of in kidney increases, so that the water loss from our body is reduced. And that there are the two critical condition we will meet when we go outside in 46 degrees. One is the higher temperature. Second is water loss. So to adapt the higher temperature, when we go again and again in sunny weather, our body color may changes due to melanin deposition. So this can be this is the body wants to take our maintain the proper temperature or its proper environment for all physiological activity, and the try to this stable maintain the stable things is called homeostasis.
Yeah, comprehensive answer, great answer, but can you give me one more example apart from temperature where we can see such very quick adaptation to a changing environment? this question is to every one of you. Apart from temperature, because temperature is the one which we like notice very quickly, right? But I want you guys to go ahead and dig deeper. What other adaptations which are like at real time scale we can observe? Any? Yeah, Mansi. Yeah, please go ahead. If you know any other example apart from temperature, where we can say that our homeostasis is being maintained by doing something or something else.
For example, the activity of enzymes. Whenever we have fever, the body temperature rises, and the enzyme do their work at a particular temperature and temperature range. So whenever we have fever, the body temperature rises and the enzyme activity slow downs. So I
No,
think
no problem. See, not at all. There is no problem with this statement, but this is not 100% accurate, right? Because on one hand, just think, even if you don't know the like deeper information or you don't know much about it, but as you said, enzymes like particular temperature, correct? You are with me till here?
yes, ma'am.
Yeah. And we are increasing our body temperature, right?
Yes, ma'am.
Because of fever. So if this first commodity likes one temperature, we are changing it. So will it increase or will it decrease? Because it was already at its optimum, right? When we are completely healthy.
Yes, ma'am.
Right? And we are changing our healthy state.
Yes, ma'am.
Functioning of the enzyme, will it increase or will it decrease?
Ma'am, it will decrease.
Yes. And earlier you mentioned it will increase. Correct?
Sorry.
No,
Yeah.
no, again. Listen to me completely. There are certain enzymes whose activity will actually increase.
Yes, ma'am.
As a consequence of fever. Because there will be pathogens because of which your body is suffering or any other illness, right? To recognize the pathogen, to process them, your system will be working very fast, right? For your system to work very fast, what you need? You need enzymes, correct?
Yes, ma'am.
So this is what I mentioned. Your answer was not completely true, but the statement through which you have started your answer should have landed you to the conclusion that the enzyme activity will decrease. But your conclusion was that the enzyme activity will increase. That is a particular subset of enzyme which will be completely dedicated in eliminating the infection from your body. Those activity will increase. But overall, ideally, it should decrease for other things because we are changing our homeostatic condition to something else.
Yes, ma'am. I totally agree. Yes.
Yeah. Okay, great. Yeah, Samadrito, you want to add something to it?
A real-time experiment, I thought that if a person met an accident, there will be lots of blood loss. When there is the loss of blood loss, there will be certain mechanism you may mention as a rush mechanism in our body, so that erythropoietin secretion increase and which will
Can you
help
observe
to.
real time?
No, I can't observe real time.
Yeah. So my question was that any other example you guys can think of when for homeostasis? Okay, I'll give you a hint. Okay, let me give you guys a like bit clever hint. Okay, what happens when what are the changes you observe in your body when you guys climb up stairs or say 3 floors all together at once without taking rest? Yeah, I guess Samadrito, you can answer. Yeah, go ahead. And I want others also to please participate in this. Yeah, Samadrito, please go ahead.
Can you repeat the question? I did not get it.
I said it is my hint, and I'm saying the question was any other activity or observation you guys can give me example of that how our homeostasis is being maintained or regulated at real time? For this question, my hint is, what happens when you climb up 3 stories all together without taking rest at every floor? So what you observe when you do this? When you don't take a lift at the end of the day, and you don't take escalators, what happens when you climb up the stairs?
Heavy respiration,
Yeah. And
sweating,
yeah. And there's one more thing which you can observe in real time and without using any instrument. So your Samadhito, your options were respiration increment and other was
sweating,
sweating. And Mansi, do you want to add something to it apart from?
Increase in the number of RBCs.
You cannot observe this, my dear, directly, right? You can
Yeah.
you pull your blood out, correct? So
Yes, ma'am. Heavy breathing.
correct. So this is what Samadhito
Ma'am,
mentioned,
I
right?
want
The
to add
increase in
one.
the go ahead.
If we experience fatigueness in our muscles.
Yes, this is what I wanted to know. So clear signs, right, that we have challenged our system, that we have given certain tasks for our system to do because we were at rest. We were at best of our capability to give our system rest and be in the homeostasis, right? So what we did, we climbed up the stairs, or say we did exercise, or we rode a bicycle, or we did trekking, we did swimming, or any physical activity which was not the zeroth point for our body. So first thing we observe is heavy breathing, right? Or respiration. We cannot say respiration because respiration is a cellular level process. Breathing is a surface level process. So we'll go with breathing option. We experience heavy breathing, like we breathe very like a frequency of breathing increases. Have you guys noticed that your breathing number increases but the length of the breath increases or decreases? When you do exercise, your breathing increases. This is a very clear fact. But the length of your breath increases or decreases? Correct. It decreases. That is why we are doing frequent breathing, right? Second was, what was second? That we perspire, right? We sweat. Correct. Why we are sweating? Anyone? Because we did
reduce body temperature,
correct. We did exercise, right? Heat was generated in our system. We want to like exclude all the heat. One way to do it, we sweat a lot. So is the heat getting dissipated from our body when we sweat? Yes or no? That perspiration is the reason, or the sweating is the reason that we are losing heat or we are regulating a body temperature. Is this statement absolutely correct or somewhat correct? Anyone? You can type in the chat box. Don't worry about it. If you're not comfortable unmuting yourself. And those who are comfortable, please go ahead. I feel like that I'm teaching to humans, not yet your replicas or avatars. Okay, so Kunithi agrees with my statement. Somewhat, Gauri somewhat agrees. I'm not a good instructor for this course. I'm not able to get my audience 100% with me. Okay. So the correct answer is that I'm somewhat correct, right? When you perspire, when you sweat, that water is not conducting any heat. It is not regulating your body temperature. It is the effect of evaporation from your body, which is taking out the heat. The moment you sweat, your surface has water molecules, right? External environment is dry. It doesn't have water molecules, correct? Again, I'm not talking about humid places. We'll get back to it. The outer environment, the external environment is dry. So what will happen? The heat from the surrounding will evaporate the water molecules from our body, leading us feeling cool. Correct? Everyone till here with me? Yes, no, maybe anything. Thumbs up if you are with me till here. Okay, 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 great. So sweating is not leading to the decrease in temperature. Sweating is the intermediate step which is required for the evaporation from your body so that the heat can be taken away from the body to the surrounding. This is the same reason when the humidity is more in your surrounding, you sweat a lot, right? Because the rate of evaporation is decreased. If sweating was the whole sole reason that you're feeling cool, then whenever the humidity is there, you sweat a lot, right? You sweat too much. So you should feel cool also, but you don't feel that much cooler. Though you're perspiring more, but you're not feeling cooler because the rate of evaporation is decreased when there is more humidity or water molecules in the environment. I hope I am clear to all of you. Yes, no, thumbs up, anything. You guys all are human. Please, response. Yes, yes, I want responses. Okay, okay, great. And if I'm boring you, I'm so sorry. I'll try to improve myself. Okay, I'm just excited about this subject. Yeah, okay, great. Without wasting much time, we'll move ahead, right? This is a same screenshot from the lectures. If you haven't gone through the lectures, please, I request you all to go through those lectures and then only attend live sessions, because then you can take the maximum advantage of it. Okay. Next question. We'll go through this section very quickly. So animals that do not have excuse me, animals that do not have internal control of their body temperatures are called bovines, ectotherms, homeostasis, feline. Even if you do not know the correct answer, with the negative approach, you can give the right answer. Great, Mansi. This is the way I want every one of you to approach. Knowing what the right answer is, half job done, right? But knowing what answer cannot be for sure is literally 90% of your job done. Oh, no, don't worry, Narendra. It's okay. See, I'll help you and maybe we can get into it. Yeah. So I think, Narendra, you might not be aware of these terminologies. Even if you not, see, look at the question. The question is asking animals that do not have internal control of their body temperature. We see one term here, right? Samadhrito, I'll get back to you in a while. Just 1 minute. Yeah. So Narendra, what I'm saying to you is that the question says we want something related to temperature. Correct? And if we look at the options, even if you do not know most of the terminologies here, we know about sixth term, right? We just discussed homeostasis, right? Homeo means internal, unchanged. Stasis means, again, static. So you can just break down the term. And even if you don't know, but like later on, like last slide only we discuss homeostasis. So C option cannot be there, right? Then if we talk about feline, so feline and bovines, these are the, scientific name for cats and cattles, right? Feline are a group or like organisms like cats. Like cats belong to feline category or feline name. For example, we are humans, we are Homo sapiens, right? Similarly, cats are called feline. And cattles like cows and all those, they belong to bovines. These are scientific terms for these animals. So bovines are your cattle, which means it cannot be related to this, right? Because we want to know the term which is associated with it. So first cannot be there, C cannot be there, D cannot be there. So what is the option left? It's obviously B. Yeah, Samadhrito, your hands are down. I hope your doubt got cleared, and if not, please ask me or raise your hand again. Okay.
Actually, I was just trying to
Yeah.
explain the question.
Okay. We'll quickly move to our next question. I've just given you a basic definition of what ectotherms are, and don't worry if you don't know it. Everything will be covered in the lecture, and if not covered in the lecture, you can always, the concepts are clearer in the lectures. the terminologies, you guys can always work with it, right? Every single book has those terminologies, but always focus on concept. Our next question is the Arctic fox. Again, Narendra, I want you to answer this particular question, okay? Because chances are no one knows about it. But how we can answer these type of questions is by eliminating that, okay, this cannot be for sure, right? So just try to attempt this question. The Arctic fox uses its dash as extra insulation when it curls up to sleep in cold weather. Yeah, great, Narendra. In every single one of you who are trying, because obviously we haven't seen, Arctic fox is not our pet, right? So obviously the correct answer is fluffy tail. Ears cannot help you to curl up and cover you all, right? So in among these options, the most reasonable answer will be a tail, right? And for your reference, this is Arctic fox. And foxes, Narendra, will belong to cat category or dog category? Dog category. Okay. So we have next question is, the processes of temperature control are centered in the dash of the advanced animal brain. These things have been covered in the first week lecture, so I expect you guys to know all this. So the first option is hemorrhage, hippocampus, hind limb, or hypothalamus. Okay, great. I'm receiving multiple answers. Most of you guys are, I'm not gonna comment for those who are still trying to answer. Go ahead, keep texting or just writing in the chat box. So obviously you guys are very smart and you guys are correct. The answer is hypothalamus. And if you do not remember from where it has been, or in the lecture where it has been discussed, if you remember when we were talking about negative feedback loop, the temperature sensing organ came into discussion. So go back to the lectures if you don't remember all this, okay? Our next question is, dash is the process of maintaining salt and water balance across membranes within the body. Is it distillation, osmoregulation, salt acid balance, or pH? Great, great. All of you guys, I'm very happy with you all. Obviously, the answer is osmoregulation, right? So here I've given a very brief definition of osmoregulation for those of you who are not clear. And don't worry, you can always ask me questions in next session. And also, as I have told, there are other 2 TAs for this particular course. So you can go anywhere and ask your doubts. There are literally no limitations for you guys. Okay, so take maximum advantage. Our next question is, dash is a medical process of removing waste and excess water from the blood by diffusion and ultrafiltration. Is it blood transfusion, blood thinning, dialysis, nephritis? So I want every one of you to answer me. Yeah, the correct answer is dialysis. And some of you who might be fascinated about this technology, which actually helped millions and millions of people throughout the world in increasing their life expectancy, looks something like this. Not something like this. This is one type of a dialysis machine only. Okay. Our next question is, any homeostatic process that changes the direction of the stimulus is a positive feedback loop, negative feedback loop, equivalent loop, or distributive loop? So even for those guys who haven't undergone the first week lectures, you can answer this, right? Just understand the question, what it is trying to ask. It is saying a process, specifically a homeostatic type of process, which changes the direction of stimulus. And here stimulus can be anything, right? It can be light, it can be temperature, it can be pH, it can be pressure, it can be oxygen level, it can be like physical stress, emotional stress, and all those things. So which are changing the direction of the stimulus. is called a negative feedback loop. Those who have answered positive feedback, a very basic difference is negative feedback. Try to understand these two words, right? Feedback is you are giving something back. I'm giving you some question, you are giving me answer. Your answer to me is a feedback, correct? So if you are enhancing my question, right, or you are answering in the direction of the question, that means you are putting something in the direction of the stimulus. I was the stimulus here, right? This will be positive because you are increasing it. On the other hand, if I'm asking you a question, that's a stimulus, and you are somewhere else, you are exploring the answers. So the stimulus direction was me, but you are going away from me, which means you are decreasing my efficiency. You are in the opposite direction of me, right, in giving answers. So you are reducing me. You are in opposite direction, which means you are in a negative feedback loop. Okay. Again, the same thing. If you want to get, I will strongly suggest you all to go through these lecture weekly by weekly. Don't try to mug it up all together, okay? Because these are very well planned and well structured lectures since week 1, all the lectures week 2, week 3 until week 12. Okay, so try to be in the loop as much as you can. Our next question is, heat can be exchanged between an animal and its environment through radiation, evaporation, convection, conduction, radiation, evaporation, convection, and conduction. Yeah, I guess most of you guys will agree with this answer, and I don't specifically need to mention why that answer was correct. Okay, good job. So our next question is, when bacteria are destroyed by leukocytes are a type of cells, dash are released into the blood. Is it fatty acid, carbohydrates, pyrogens, or pathological particles? Excuse me. Yeah, I think this was well covered in the lectures also, right? So pyrogens are small molecules which causes or which leads to the fever. They can be peptides, they can be other type of molecules. They uh, uh, like start the cycling of us getting fever. Or pyro means fire, right? Something related to temperature, fire, heat. Wherever you see a term pyro, it will always be associated with temperature, heat, fire. Okay, so always remember this. And our last question for this session is, dash. I guess most of you should answer this question. And one hint, India stands among the top nations who is suffering from this. Yeah, it's a sad truth. Yeah, I guess all of us knows someone who is suffering from this critical disease, right? And still they don't take it seriously. I feel sad for them. Yeah, it is diabetes, right? So this is all from my side.